just came across a 4-page document called Post Notes, which the government releases every year. They're 4-page documents on areas which the UK government thinks we should innovate in science and technology. And one of them in 2018 was on the microbiome. And there was just this one line. So I was reading about the microbiome and I was saying, we need to really innovate in the microbiome. Here's the reasons why the microbiome is interesting. And it's a really interesting document, looking back at it now. But there's just one line in that note where it just said, the breast milk seems to have microbes in it, which is interesting. And babies not breastfed are more likely to get, autoimmune diseases. And I was just like, what?
Yep.
That was
a brain explosion.
Yeah, because I just assumed it was sterile. I didn't realize that there was a microbiome in breast milk.
Right.
I mean, now there seems to be a microbiome
basically nothing sterile.
Yeah, exactly.
There's microbiome in everything, including blood.
Yes, yeah.
Which is another mind-blowing thing. Yeah. But anyway, sorry, yes.
Yeah, so read that.
That blew your mind.
That blew our mind. Then we're like, what is in this breast milk microbiome? And there have been a number of papers published, but it wasn't included in the Human Microbiome Project. And the level of detail.
And for the listener, that was a big deal, wasn't it? That
Yes.
was funded by the National Institute for Health in the US, focused on trying to map the microbiomes of the body, essentially, right?
Yeah.
And it didn't include breast.
No.
Wow. Okay.
So the level of detail just wasn't that high of exactly what bacteria are living in this breast milk microbiome. How do they get there? How does it differ? There wasn't a lot of detail at that point in 2018. And so we just thought, well, you know what, this is classic women's health in particular being overlooked, underfunded. Let's see if we can innovate in this space. And so we just came with that idea. We weren't really planning on taking it forward, but we just got such good feedback from that competition. We just sort of looked around, and actually we were a team of 5, and we were all doing PhD students. We were all doing PhDs, and me and my 2 co-founders that took it forward were the ones that sort of were like, we stayed up really late the night before the pitch. We realized we worked really well together.
Okay.
And the other 2 amazing scientists now, both really good academics now. Yeah. But they decided, for them it wasn't.
They wanted a different life.
But that was fine. We're still really good friends with them. And like I say, they're very successful academics in their own right. But yeah, we took it forward with 3 of us, and we just started doing competitions.
How was the pitching competition though, by the way?
Oh, the first one?
Yeah.
Oh, awful. We lost. We didn't even get through to the next round.
Why? What happened?
I don't know. No, there was just another company that was better than us. Also, interestingly, but they didn't go forward with it though. They didn't win either, which is sad.
That's sad.
But yeah, they also, the one we lost to, were doing acne treatment using microbiome.
Okay, topical.
Which is very relevant, to be fair.
Very, very relevant.
And I don't think they went forward with it.
I mean, acne is driven by the microbiome. Yeah. It's just bugs that are getting caught in the glands. I can't remember what they're called. Sebaceous glands or something like that. Yeah. Fascinating.
So we didn't win, but that was our first taste of failure, but also first taste of actually excitement.
And at that point, were you already in your head fine at the idea of, or whatever, actually doing this alongside your PhD? If something was to come of it, you were going to do it at the same time, because that's a huge thing.
It's one of those things you do a little bit. We're like, oh, this is an idea. Let's just see how far we push it.
Yep.
Just push it a little bit. We'll do another competition. Why not? We didn't win that.
What was the original pitch idea? Can you remember?
The original idea, and maybe we will do this one day, was to sample women's breast milk, look at the exact microbial composition in her milk, and then if they're missing out on any key species, we'd supplement to give a very personalized product.
So if you now supplement
with the bacteria back from our culture collection,
and it would find its way into their breast milk.
Oh, sorry. Then we should mix it with her milk and then feed her infant.
That makes a lot of sense. Okay, that makes a lot of sense. Got it.
That was our original.
So I was like, how does it get there? But how does it get there originally?
Oh God. Everyone asks this question, and no one's entirely sure.
No one knows.
There's two theories.
Okay.
Theory number 1 is that it just comes from the infant mouth. So when the baby's feeding, it just sort of, the bacteria in the baby's mouth goes back into the ducts and it crawls up. And there's been a bit of evidence for this, if sampled babies' mouths.
But I suppose, just to push back a little bit, could it not be that it's the other way? Okay, right.
Yeah. So that's theory number 1.
Yeah.
But theory number 2.
Interesting.
Is a thing called bacteria translocation, and that's when bacteria hitch a ride on an immune system cell and sort of travel through the lymph symptom to the ducts. And there's been there's been evidence both for this. There's been evidence where they made these poor, sad, germ-free mice. These are mice which haven't been exposed to any microbes, and they fed them a probiotic, and then they got them to have baby mice, and they found the bacterium that they fed that mouse showed up in the mouse's milk. So, and so, you know, it's got a lot of evidence with it.
It's like a sci-fi thing.
It is.
They hitch a ride on an immune cell and then go, yep, this is where I need to be.
Yeah. Yeah.
And that might be some sort of incredibly conserved evolutionary thing that just happens with all humans, which again is another one of these, my mind's
There's just so much we don't know though. But, you know, there's so much more research needs to be done on this. And it's also, it's one of those things where, you know, if it's really difficult to research it. And, you know, our techniques for culturing and assessing a microbiome are still pretty new. So sometimes people ask me, oh, do you think if a mother was a C-section baby herself, does that mean that she's passing on, she wasn't exposed to the first vaginal microbes of her mother? Does that mean that her infant microbiome won't be as good or will be less diverse? And you've just got to be like, we haven't been able to study it.
Right.
You know, so it's really exciting. You know, as we start to do these longitudinal studies, we'll get more answers on that. But right now, these techniques weren't really around.
So you're talking about the analytical toolkit that we have.
Yeah, absolutely. So,
and it has come on a long way though, hasn't it?
Oh yeah.
I mean, we were up until relatively recently trying to grow everything on plates.
Mm-hmm.
That's all we had.
Yeah.
And microscopes. Yeah. But now we've got next generation sequencing technologies.
Yeah, it's changed everything.
PCR, you know. So, okay, cool. So it started to snowball. And it's funny, as you're telling the story, I'm thinking, God, it's the same as me. It was a super exciting thing.
Yeah.
And I think if you'd shown me a crystal ball and said, yep, in 2023, you'll be employing almost 70 people, you have an MHRA license, you'll be just about a phase 2 study, I'd be like, what? Surely not. Like that's, I'd be like, that's mad. Yeah. That's insane. Yeah. And it sounds like that's what's happened with you. It's just like, it's trickled and then it's just become this huge thing.
Yeah. Yeah.
You're going to get to the point probably in the story where it became almost unmanageable to do the two.
Yeah, it was pretty unpleasant at times. Well, do you know, it was amazing. So we managed to get a very little amount of money. So we managed to get 50K to work on our first sort of proof of concept. We started collecting milk. And with our PhDs, we were really lucky actually. So me and my two other co-founders. So I'm a...
Can I ask you, sorry, just I'm sorry to interrupt you, but you were talking about the original idea.
Yes.
And then we were going to get onto what it became. And that's probably what you raised the 50K for, is that right?
Yes.
Okay, so what was then the idea that it became that you got some money?
Well, it was when we realized that the logistics of trying to do a personalized medicine approach for microbiome was just going to be an absolute nightmare.
One day.
One day.
So cool, right?
Very cool.
Yeah. One day that would be the thing to do.
And we started looking more into the different babies which are most vulnerable to having dysbiosis, so an imbalance in those beneficial bacteria in the gut. And we realized there's three different sort of camps of babies. There's obviously the babies that aren't given any breast milk. So actually 40% of the first bacteria in an infant's gut come from breast milk. So it's a huge input of these first beneficial bacteria. So of course you'd think, okay, formula-fed babies, obviously they're missing out on breast milk. So they're missing out on these first really important
colonizers.
Early colonizers, yeah. It's been estimated it's about 30% from your birth delivery as well. So it's 30% from when you're born, from if you're born naturally, you're born, you know, from your mother's vagina, and you're coated in all those microbes. And then of course, if you're lucky, maybe she'll have a little poo while she's in labor if you're lucky. So you might get some of them as well. Swallow some of those bacteria. And then the next seeding is from how you're fed. Wow. And then there's going to be about 30%, which is just...
But just think about
a dog.
You know, if you have...
Skin.
Two people or two babies, one's born through C-section and bottle fed. The other's born through normal vaginal delivery or just vaginal delivery, whether it's forceps or not, and is breastfed.
Yeah.
There's a 70%.
Yeah.
All else being equal.
Yeah.
70%. Yeah. That's huge.
Yeah. And they are much more likely to have different diseases like asthma, obesity is one, diabetes. But interestingly, they can also be absolutely fine. And that's what's so
as is always
thing about microbiome, because you can't, their genetics, or maybe their dog was really good in the house that they had, and it's really something that we're spending a lot of time doing.
Dogs are a thing though.
Yeah,
I know it is.
Yeah. Well,
dogs influence your microbiome for sure, right?
Yeah, I mean, I did a parasite course in my undergrad about dogs and cats, and I don't know, it's really put me off. But you're right. They found that babies that are exposed to dogs growing up have more diverse microbiomes in early childhood.
Yeah. But, incredible.
But yeah, sorry. So where was I going with this?
Really interesting. So you were talking about how the original idea wasn't viable because it was too difficult to scale.
Yeah.
Because logistically, you'd have to get a sample from every single mother, figure out what it's missing, using a variety of tools, and then also replace what's missing, which might be different from a mother to mother basis, and that's a different product for every person.
Yeah.
Okay, so that's not feasible at the moment.
Yeah. So that's when we started looking at, okay, well, let's if we want to refine this a bit more, what are the different babies that actually are at most risk? And it's if you're not given any breast milk because you're missing out on that those beneficial bacteria, formula-fed. The next camp is if you're born C-section, which we've touched on now. So C-section babies, they're missing out on those first sort of vaginal and GI tract microbes. And then the other group baby, which are the most vulnerable, arguably, are preterm babies. So if you're born too early, it's really difficult, firstly, for mothers to produce enough breast milk because the milk's not ready to come in.
Yeah. Physiology is not sort of developed enough yet because of where they're at in the whole process of having a baby.
Also, these babies are often on very high levels of antibiotics, as is the mum as well.
Right.
And as a result of that, they have very, very poor microbiomes. And to be honest, their stomachs also their guts aren't developed properly. So often they've got a leaky barrier, and the tight junctions in their guts aren't close together, so things can sort of slip through to their bloodstream. So they're very vulnerable group. And
how important do you think tight junctions are and leaky gut is?
Well, why are you asking that? It sounds such evil glint in your eye, James.
No, I mean I have my own view. I don't want to. I don't want to influence your thinking.
No.
There's no right or wrong answer to this, by the way. But
so, I mean,
evil glint. I'm not plotting or scheming anything. I promise. Yeah.
So I think with these preterm infants, it's fairly important because it's just undeveloped. So the crypts. I'll just smash the mic then. The crypts aren't anywhere near as developed as well. They're sort of stubby. You're meant to have lots of nice long. You probably know what the term is. I always forget it.
They're called
crypts of
crypt abscess. No,
in your gut, high surface area.
Yeah. I know exactly what you're talking about.
Yeah. They're just not developed very well, and it means if you do get a nasty opportunistic bacteria pathogen coming in, it can just very easily just slip through those gaps and go into the blood. So I think for preterm infants it is pretty important.
I think I totally agree with you, and I don't know why I was making an evil blend. I think they're important for everybody.
Yes.
I mean, for adults, arguably just as important or more important. I mean, when that barrier starts to break down, then they do translocate and influence local and systemic inflammation, and that kind of leaky gut, quote unquote, has been linked to all manner of problems, both in the gut and beyond the gut. So, figuring out how we can sort of get them tighter again through microbiome modulation is a key area of interest for me, for sure.
Yeah.
And I hadn't appreciated that it was a big thing in preterm babies, but it makes total sense. And it's probably the fact that they're preterm, and probably the fact that they're getting loads of antibiotics.
Yeah.
Right? And it's just creating an entirely not normal situation or environment for them.
Yeah, absolutely.
So, and there's a lot of C-sections and a lot of preterm babies, right?
Exactly. Yeah, it makes it even harder for them. So, I mean, there's a few diseases because of this, but preterm infants are particularly vulnerable to.
Yeah.
One of them being necrotising enterocolitis, or NEC, which is a very unpleasant disease. So actually, I should say as well, about 10% of births are preterm, and then roughly about 10% of preterm infants will develop NEC at some point. And this is a really nasty disease. So like I say, it's when an opportunistic pathogen comes in, enters the bowel, and the immune system, which hasn't really been trained how to respond appropriately, responds with really high inflammation, and then you get necrosis of the lining of the gut.
Death.
So death. Yeah,
that's a bad thing.
Very bad thing, because then you get leakage of the gut into the abdomen, and it can have really high mortality rates.
Fatal.
Yeah.